But now we've given you the overview, we'll next be moving into the specifics and showing you examples. So with that, I would like to introduce Dr. Randi Vita to present Accessing the Data, Query, Reporting, and Examples. Thank you very much, Randi.
Hi, I'm Randi. I'm going to talk to you about how you can use the query interface in a little bit more detail. Alex already did go over it in general, and then I'm going to walk you through how we report the data, and I'm going to use one user example to do this. So this is the homepage, as you've seen, and there are these 6 main key search panes, and this is how 99% of the queries that are done in the IEDB can be done. You can query almost everything you need to on this homepage, and the next 7 slides will show you a little bit more detail about how each of these panes work, with the epitope source having 2 slides because it has 2 search interfaces. So starting up at the top at the epitope, this is one of the key main ways people come to the IEDB and search. They may have a peptide that they have just predicted and they want to see if there's any data for it. So if you want to search for a peptide, you can enter that linear sequence in this box. But I want to encourage you to realize that other authors may have studied a slightly different peptide. It may be 1 or 2 amino acids longer or shorter. It may have 1 or 2 amino acids different from what you have just predicted or you want to test. So I encourage you to use these blast matches where you can do 70, 80, or 90% blast matches to your peptide of choice to get a broader search results. So always do that first and do a broad search. And then if you have a lot of data, you can narrow it to be more precise after you've got your results. Also in the epitope search pane, you can limit your search to only linear, discontinuous, or non-peptidic epitopes if you want to do that. Then the next search interface is the epitope source. So this is if the epitopes are natural, as that question asked, they will come from some organism and possibly some larger antigen. Some of our non-peptidic structures may not come from a larger antigen, they may be the antigen itself. But if the epitope is natural from an organism, you can find that organism here and you can just start typing in the search box, and all of our search boxes have autocomplete. So all of the hits that are similar to what you've typed that we have data for will come up. If you're looking for an organism and we have no data for it, that means there may not be any epitopes published yet, but we add new data every single week. So if it's something like SARS-CoV-2, at first, at the beginning of the pandemic, we had very little data, and then suddenly we have an awful lot of data. So if it's something that people are actively researching, you'll see that data set grow over time. And then the next part of the antigen source pane is the antigen itself. So this would be the protein that the epitope comes from, if it is derived from a protein. And you can just start typing just like with organism and it will autocomplete. But I also want to point out that some protein names are present in many, many different species. So for example, if you start typing capsid, you're going to get every virus that has a capsid. So it might be just easier and quicker to just search for the virus you're interested in. And then when you get to the results, I'll show you how you can quickly narrow to within all the proteins that we have data for that organism on the results page. And then the last search pane on the left side is the host. This is whose immune response is being measured. Is it a human, a mouse, or non-human primate? But we also have all other vertebrates. If there's any published adaptive immune response in that species, we'll have it. So we have birds and fish and many other mammals. And with all of the search boxes, if you see a find button, you can click on it and it will open a browser tree, and that lets you navigate to see what sort of data we have. So you can click open these folders and see all the different birds and fish and mammals that we have data for. And again, as we add data every week, you may see new species show up over time. And then now on the right-hand side at the top, the search pane is assay. So the IEDB is divided into T-cell, B-cell, and MHC ligand assays as the broad categories, and you can check and uncheck these boxes if you want to limit your search right off the bat. And we also have default positive checked, and we found that was because most of our users want to see the positive data, but we do include negative outcomes also. So if you do want to include those, you can check it here. And then you can autocomplete by typing, or you can click the find button and browse the assay tree to see all the types of assays that we have in IEDB. And then the next search pane is the MHC restriction. So for T-cell assays, if the authors tell us some restriction information, we will capture it, and we will capture whatever that author tells us in that paper. So if the author only narrowed the restriction to class 1, that's what we'll capture. And if you only want to get class 1 data back, you can hit this radio button and you will only get class 1 data. But we also have a finely defined MHC restriction when the authors provide it. So you can open the tree and browse through it and find all of the restrictions information we have to date. You can also start typing, but because MHC nomenclature is kind of complicated with dashes and asterisks and colons and slashes, it might be easier to navigate through the tree or to click on one of the high levels and then narrow it once you get to the results, because you can always narrow your results later. Then the last search pane on the homepage is the disease. And this is the disease of the host at the time their T cells or antibodies were being tested. So did the host have an infectious, allergic, autoimmune, transplant, or cancer disease that was relevant to that adaptive immune response? Even if they've recovered from it, if it was relevant, it will be captured. And like the other search boxes, you can type and it will autocomplete, or you can click the find button and see the tree, and then you'll see all the different types of diseases we have. And the value of these trees is you can search on high levels, like you can search for just all allergic diseases, or if you want to find a specific allergic disease, you can navigate through the tree and find just the diseases you're interested in. I also wanted to point out that we have animal models of disease, things like EAE, and any new disease that comes up in the literature that's relevant, it'll eventually end up in the tree as we add that new data. And so this is the example query that I'm going to show, and this is how do you see the differences between B and T cell responses in SARS-CoV-2? So since this is about the epitope source, the organism, I just go to the organism box and start typing, and it autocompletes. And the very first choice is SARS-CoV-2, so I select that. And then what you see is a 1, and this is because many of these fields are multi-select. And if you wanted to select a few more organisms to query on, you could add them, and this would change to 2 or 3 or whatever you want to pick. And you can pick as many as you want, or as few values as you want from all of these search panes. You can build a query. I could now limit it to just humans in class 1 and only humans who had a disease. I could say which specific disease, or you can go very broad and not pick anything and just go to the IEDB homepage and hit search, and then you'll get all of our data. So once you figured out all of the search parameters you want to add, you click the green search, and then you get to the results page. And I'm going to walk through the details of this page because there's a lot to it. So when you first land on the results page, the current filters up at the top will show what your query was, and you can X these off and then research, and your search will become broader again. It also helps to remind you of what you may have clicked on. And then we also have this pending filters over on the left side, and here's where you get a few additional search features. We want to keep the main search parameters that most people use in the homepage, but we also still want to make available to you additional search features because we capture an awful lot of detail, and it would be a shame if you couldn't search on it. So I'm going to show you when you first land on the results page, you may be on the default T cell, B cell, or MHC filter options view, depending on what your query was. If I had put in my query that I only wanted T cell results, I would have landed on the T cell results page because it's smart enough to understand I'm only looking at T cell data. But because my query was really broad, it's just about this organism, I landed on the default page. But even on the default page, we get additional search features that we didn't have on the home page over here on the left. So I'm going to show you how those are. So the very first pane at the top is the epitope, just like on the home page. But now you additionally have the ability to search for 3D structures if you only want data where there is a known 3D structure. And then also if there were any post-translational modifications done to the epitope residues, you can search for those here. And then in the epitope source box, in addition to the organism and antigen that were on the home page, we now also have the ability to include related structures. So these are analogs, mimetopes, and neoepitopes of whatever my search parameters were. So there was a question about, are all epitopes from an antigen? So these epitopes may be artificial. So if you're an analog, so it's a very similar peptide to a SARS-CoV-2 peptide, but a few of the amino acids have been changed intentionally, so now it's no longer natural. If I wanted to include those in my search results, I could check this box, and I would get also analogs of SARS-CoV-2 epitopes. And then, as Bjorn mentioned, we do have a receptor search interface because we do capture TCR and BCR sequences whenever the authors provide them. And we capture whatever information the authors give us, whether it's a full-length nucleotide or protein sequence. If it's a CDR1, 2, or 3 sequence, we'll capture however much they give us or however little they give us. And it is searchable, and you can search in the high levels of TCR, BCR, or you can search for a particular CDR1, 2, or 3 or full-length sequence by entering that sequence here. Say, for example, if you've sequenced a CDR3 and you want to see if there are any known epitopes for it, you can do that in this search box. And then lastly, in addition to the panes available on the homepage, we also add the reference. And so this is where that data came from. Most of our data is from journal articles, but we do also have submissions. If you want to look for a certain author, you can put your own name in here and see if you have any epitopes from your papers. You can search for the title and then the date range. We have all data from the beginning of PubMed until about a week ago. So if you want to limit your search to being very recent, or if you include everything, and then if you have a particular paper of interest and you want to see if there's epitopes curated for that paper, you can put the PubMed ID in. And then if I had landed on the filter options for T-cell, or if I chose to go to that tab, you get a few different things that are different from the default view. So the default filter options, it was the receptor search pane, but on the T-cell view, it's only searching TCRs now. So any of the choices you put into this search box, you'll be only searching through the TCRs. So it's the alpha and beta chains that we have data for. And then when you're searching in the assay search pane, you're now only searching T-cell assays. And so some of the more common T-cell assays are pulled out as radio buttons to make them more quickly accessible to you. And then we also add the feature of being able to search for direct ex vivo detection. So these are cases where the T-cells were taken from the host without any prior re-stimulation in vitro before the experiment. So they're direct ex vivo experiments. And then in the MHC restriction box, we add two new features that are not available on the homepage or in the default view, and that's the resolution and the evidence. So the resolution is how well defined is that MHC restriction? Do we only know the class level? Because we will capture the class level if that's all the authors give us. But if the authors give us one of the chains or both of the chains for the restriction, we will capture the fullest detail that they give us. And some of our users we found really only want highly defined restriction. They don't want to look at the class data. So you can make that distinction here and add that to your query. And then the last thing added to the MHC restriction box was the evidence. And this is the evidence codes that you can choose from. And this is also because some of our users only want to know MHC restriction that is very definitive. And we let our users decide for themselves what they think is definitive. In some cases, a single allele present, say like a tetramer assay, but other authors may only do MHC binding predictions. And if you don't want to include MHC binding predictions in your results set as a way to determine restriction, then you can check the ones that you think are valid, and it'll limit your results set to only those cases where the restriction was defined in the manner that you think it should have been in order to be relevant to you. And then we also have the B-cell filter options. So if you go to this view on the results page, when you're searching the receptors, again, here you're only searching antibodies and BCR sequences now. So you're searching heavy and light chains. And when you're in the assays, you're only searching B-cell assays. So this tree would only show you different types of B-cell assays. And we add the ability to narrow by isotype. So if the authors tell us the isotype, we do enter it. So you can now narrow your results to IgE, IgA, IgG, or IgG1, whatever information the authors give us, we will have that. And this is the last of the filter options, the MHC filter options. So here, just like the others, we're only searching through MHC assays, with the two main groupings being the MHC binding assays and MHC ligand elution assays. And then in the restriction, just like with the T-cell, we also add the ability to search the resolution and the evidence for that restriction. Then going back out to the results page, it is organized by these tabs, as Alex had mentioned, and there's these main headers with the epitopes, antigens, assays, receptors, and the references. And on the epitopes tab is usually the default where you're going to land. And each of the tabs has its own set of header rows that is sortable with these little arrows. You can click on them, and it's default sorted by the row that has the most data. And many of these tabs will have a column in the far left with an ID, and this ID takes you to details about that row. So this is the ID for this epitope. And if you click on it, you'll go to the details page for the epitope, and it gives you a summary about all of the data we have for that epitope. And these will change over time because some epitopes continually get studied again and again. And this one, this glycoprotein epitope happens to be studied an awful lot from 37 publications. It's very positive. Every time it was tested in a 2-1 assay, and then here are all of its T-cell assays, and it's a generally quite positive epitope. But I also wanted to add that this is an infectious disease epitope, and for allergy and autoimmune epitopes, we do also curate the healthy controls. So say an epitope that is important for diabetes, it may be positive in only the diabetic subjects and negative in all of the healthy controls. So it may look like it's positive half the time. And that's just because we capture both patient populations, healthy and diseased. And then going back out to the results page, each of these results pages have these inline filters, and this is how you can narrow your results quickly on the fly. So you can do the broad search, get all the data, all the epitopes from it that meet your query parameters, and now you can narrow it. I could narrow it to only see data for this epitope. I could narrow it now to only see data for spike glycoprotein if I wanted to limit it to the specific protein. And each of the tabs will have similar, these funnel signs that let you narrow the results. And then each of the pages let you export the data, and that's over on the right-hand side. And when you click on the export button, you get options, and Kelly Wheeler is going to give a talk a little bit later about some new features we're adding to our exports to improve them and add more value. And then the next tab over from the epitopes is the antigens tab. So these are the proteins that are the sources of the epitopes that meet the search criteria. I had all of my search criteria SARS-CoV-2. So these are all the SARS-CoV-2 proteins for which we have data to date. Some viruses have certain proteins that are very highly studied for good reason, because they're the ones that are interacting with the immune system. So you can see spike glycoprotein and nuclear protein have an awful lot of data. But some of the other proteins may have no data. They won't be in this view at all if they have no data, or they may have very few studies. They may have been studied a little bit and then no one pursued it. And again, this always is reflective of the literature, what's known to date. And then the next tab over from antigens is the assays. And so this is the bulk of the IEDB. These are the experiments. This is where all those epitopes came from. They were tested in all these different experiments. And this tab is divided into the subtabs of T cell, B cell, and MHC. And each of the subtabs has the same header row that's sortable and default sorted. And then there's these funnels again. If you now wanted to, on the fly, limit your results to only human data, you could hit this little funnel and it would do that. If you only wanted, if you saw a paper you're very interested in, you only wanted to see the data that came out of that paper, you could hit the funnel and do that. And then just like with the epitopes, there's an ID column. And this ID takes you to details about this row. In the results page in these assay tabs, we're showing you a kind of an overview summary of the experiment, but we capture way more fields than is shown. We capture up to 400 fields for a T cell assay, and we can't show that all here on the web page. So if you click on the details, or when you export the data, you get all of the details about that row, and they may be very valuable details for you to see. So I'm going to click on this assay ID and show you those assay details. And so when you do that, the details pops up either in a new browser window or tab, depending on how your browser is set up. And it's a long web page, and up at the top, the first thing you see is about the reference, where the data came from. You can always go back to that paper and look at it yourself. And then the next section is always about the epitope, because all of the data in the IEDB is about an epitope-specific adaptive immune receptor. So there must be an epitope for that receptor. And in this case, this is the epitope, and these are the link outs. And the place in the paper where we got the sequence will be shown in the location of data. So we got a supplementary table S5, so you can go there and look. And then the next section is about the in vivo immunization. Who was the host and how were they exposed? So in this case, it was a human who had COVID-19, but they had recovered prior to the experiment. And this disease was caused by SARS-CoV-2. And then the next section is about an in vitro immunization, if there was one. And in this case, there was. They took PBMCs from that human, and we stimulated the spike glycoprotein, the whole protein, prior to the experiment. And then what was the experiment? It was a tritiated thymidine proliferation assay, where they used a T cell clone. And if the authors give us a name for the clone, we will add it. If the authors give us the sequence for the TCR, we will add that. And then lastly, if we have MHC restriction information, in this case, we only have it to the class level. And then what was the antigen? And here the antigen was a sibling. It was a SARS-CoV-1 S protein. So it's important to note that the immunogens and antigens are not always the epitope. If an adaptive immune receptor binds to anything else or shown to proliferate or recognize any other antigen in the paper, whether it's monoclonal antibody neutralizing the whole protein or a T cell line proliferating to a different protein or other cross-reactive epitopes, we capture all the context as long as that receptor was epitope specific. Then going back out next to the assays is the receptor tab. So this is where all those TCR and BCR sequences are. We have the tab for each. And like the others, we have a header row and the IDs, and you click on the ID and get more information about it. You can also export this information. And when you click on the ID, you get a detailed summary about that CDR3 sequence. So we group our different receptors that have been added by all sorts of different papers having different epitopes. We group them together based on their CDR3 sequence, because different authors may report different bits of information about that same CDR3 sequence. So in this case, these different authors never sequenced the alpha chain, so we have no information about the alpha chains here. But if they did, we would also capture that data. And for this CDR3, it's very interesting because it was shown to bind an influenza A epitope, also a yellow fever epitope, and a SARS-CoV-2 epitope. So that same CDR3 bound a couple of different epitopes from a couple of different viruses. And then the last tab on the results page is the references. And so Alex had mentioned this in an answer to one of the end users about how do we get to the reference information. And all of the data, when you export it, is linked back to the paper it came from. All of those assays will tell you what the publication that assay was curated from. But this reference tab, if you exported this, you would now have a list of papers that have epitope from SARS-CoV-2 in them. So that would just give you that. But you also narrow to a particular paper. You can look at the details for a paper, and you can link out to look at that paper itself in PubMed. So back to the query that I was trying to illustrate, which was the differences between B and T cell responses for SARS-CoV-2 epitopes. So if you go to the assays tab, you'll see there all of the T cell assays and all of the B cell assays for SARS-CoV-2 epitopes. And you could export those and just be done with that. But there's additionally things you can do to narrow the results and look at the differences. Is if you go to the assay search pane, either on the home page or on the results page, you can uncheck B and MHC, and now you'll only be looking at T cell data. So this is the 3,000 T cell epitopes from SARS-CoV-2. And similarly, if you uncheck the T and check the B, these are the 9,000 B cell epitopes from SARS-CoV-2. But it's entirely possible for different authors or even the same author to test the same peptide in both T cell, MHC, or MHC ligand assays. It's more common to have overlap between MHC and T cell, but you also do sometimes get B cell and T cell assays being studied, especially when authors test overlapping peptides. And then lastly, I want to show you, Alex had mentioned this, and later today, Alba will give a more detailed talk about how you use Immunome Browser and all of its features. But when you first go to the antigens page for the first time, you'll have this little pop-up window about our Immunome Browser. And that's because it's one of the coolest features of the IEDB to really help you visualize and see the differences between different types of data. And if you click on the Learn More, you go to a help article about it. Alba will give you all sorts of details. You can uncheck this if you don't want to look at it. But if you're trying to get to the Immunome Browser, it has this little graph icon, and it's next to the protein of interest. It's taking the data and plotting it to that protein. So if I click on this icon next to spike glycoprotein, I'm now looking at the results of my query plotted along the length of the spike glycoprotein. So this is where there were epitope assays that were positive in blue and negative in red. And if there was a region of the protein that didn't have any experiments, it would be blank. If there was a region that was always negative, it would only have a red line. And the differences here you can see between B cell and T cells, the blue spikes are in different locations. You can see that they often will look very different. And this is always relative to what your query was. If you wanted to do the query for just humans who had COVID, you will get slightly different results. In this case, I just did all epitopes. So these are not specific to humans or infected humans. And then one last step was I separated CD4 and CD8 T cells by doing two different queries by picking the MHC restriction of class 2 or class 1. And you can see it's quite different plots where the peaks are for the CD4 versus the CD8. And there's this one prominent peak here for the CD8s. And I bet you that's where that epitope, that A0201 epitope that has the most data, is probably right in that peak. And I also want to show you to pay attention to the scale. So there are 100 epitope assay count scale for the CD8, meaning there's a lot more data, and only a 40 epitope assay count for the CD4, so a little bit less data. And this again always reflects what your query was and then what has been published so far in the literature. I think that was my last slide.